Okay, we're on the second to last type of marker that we are going to discuss in this module, and here we focus on what are called SNPs. And the basis for this particular type of marker is that closely related individuals have very similar DNA. And so that's shown here in this image, which is actually one I showed in the last video. These are DNA sequences from 5 different species of freshwater mussels. Remember, each one of these asterisks here indicates where the nucleotides are exactly the same, even across these different species. And when we look at this top line, we see that most of these DNA sequences, most of the nucleotides are actually the same in this sequence, even across these different species. And if we were looking at different individuals from the same species, they would be even more similar, even more asterisks. I need to change my lectures, so I need to say that word less. And so, but the overall idea is that because many individuals have such similar DNA, a better approach than looking at all of these DNA nucleotides and all of the DNA sequences is to instead focus only on the nucleotides that are variable, or the ones that are different between the individuals or between the different species. And so that's where SNPs come in. So SNP stands for single nucleotide polymorphism. And these SNPs are those variable sites. So that's illustrated here with 3 different individuals. They have a certain DNA sequence that we're looking at that can be either in coding or non-coding regions. So that's an important distinction to make. So you can find SNPs in regions of the genome that are coding and actually result in a gene and protein product, or non-coding regions that do not result in a protein product. SNPs can be found in either. But here in this cartoon schematic, we see that at this particular nucleotide site, which has SNP indicated there, the nucleotides are different for these 3 different individuals. Whereas for the rest of the sequence, everything is the same. This is one of those targeted variable sites that we can use as a marker to inform our studies. That's a SNP. If we want to think about how this particular marker compares to other markers we've talked about so far, like microsatellites, for instance, SNPs are less variable than microsatellites. So remember, we said before that microsatellites are really variable. We use that example with the Pyrenean desmin. SNPs, though, are less variable. So here in this image, don't worry about this part, but we have individual sequences. So each line is a different individual. And in these gray boxes here, we have identified regions that are variable. So these are 3 different SNPs. If we think about it, there are only 4 different possible nucleotides, right? So at any given nucleotide position, there are only 4 options for having a nucleotide there. And in fact, most SNPs are what we call biallelic, which means they only have 2 options. So this is a SNP that would be considered biallelic because all individuals have either an A or a G nucleotide. Here, all of these 3 listed are biallelic. They only have 2 nucleotide options. So SNPs are generally less variable. If we think about microsatellites, for instance, though, microsatellites, remember, are giving us a number of repeats. And so we can have up to 10 plus different number of repeats. So whereas for SNPs, we only had 4 nucleotide options, here we can have 10 plus repeats. So that's one of the reasons why microsatellites are more variable than SNPs, because there are more options available for how many different repeats, in this case of that GA nucleotide sequence, you can have. However, even though SNPs are less variable than microsatellites, they are much more numerous. So for microsatellites, generally there are thousands of them in the genome, whereas SNPs, as you may have guessed from my Austin Powers meme, there are millions of them. So even though SNPs are less variable, they are more numerous throughout the genome. Millions, in fact, millions. And what that means is that even though SNPs are less variable, because they are more numerous, they can be really useful for studying adaptive genomic regions. So remember, back one of those early videos in this module, we said that an adaptive marker is something that affects an organism's fitness in its environment. And if you note what I did here, I have this little dollar sign. So SNPs are more expensive than microsatellites. So that's one thing to consider. But they are more commonly used for studying adaptive genomic regions. And one of the reasons for that is you often have to start with a huge number of SNPs in your data set to be able to actually find any that are adaptive. I'm showing another picture of my snails. Don't worry, I have lots of different snail picture options, so I will not run out. And this is actually a video. So if you download that PowerPoint that I have for you, you can watch a very riveting video of a snail moving around at a snail's pace that you would expect. But SNPs are really useful for studying adaptive genomic regions because they're so numerous. So in this particular study that I've talked about a couple of times now, where I was looking at northern and southern populations of snails, I had to start with roughly 1,600 SNPs overall throughout the genome that I identified, and only about 30 of those were found to be adaptive. So that just goes to show you have to cast this really, really broad net and identify a lot of SNPs within the genome to be able to find any that are adaptive. So this is one example from my personal research. The example they use in the book is that they started with 200,000 SNPs. This is in a study with Arabidopsis, I believe, and they found only 4 were associated with fitness in these different field conditions. So that was something like 0.2% or something really, really tiny. And so that's why it's beneficial to use SNPs when you're studying adaptive genomic regions, because you have more options, you have many more markers to choose from, and then you can whittle that down and have a better chance of finding some of those very, very few ones that are actually adaptive. And we're talking about each of these different markers separately in their own video. So this is the SNP video, the next one's going to focus on high throughput sequencing. But really, a lot of these different techniques and markers can overlap a little bit. And high throughput sequencing, which we've talked about in the previous module, is actually one of the ways today that can be used to identify SNPs. And that's actually what I used in that study I was just referencing. And the study I've been going back to throughout this module, where we had those conflicting results between my study and the previous paper, I actually used high throughput sequencing to identify SNPs in my data set between Northern and Southern California snails. So I've shown this image before, but again, if we're interested in looking at SNPs, what's highlighted in these yellow boxes here, we can see that there was one identified SNP in snails. I can draw them again, from Northern California versus those snails from Southern California. So high throughput sequencing, which again is that massively parallel sequencing that gives us lots and lots of data back, that can be used to identify those large number of SNPs that we need in our data set to be able to identify those very few ones that are actually adaptive.